We're often tempted to describe soils as inanimate collections of grains, and then we will let the water flow through them, and we can describe that, and we can repeat the same experiment over and over, dry the soil in the oven, and we're good to go, and we now have a full description of that material. I want to bring forward a few experimental results to show perhaps the limitations of that sort of vision of soils. So in this case, I'm showing some experimental and modeled results for sand, which is being colonized in time by bacteria. So what we're looking at is 6 days worth of colonization, and the spatial scale on this is about 30 centimeters. That black window is about 30 centimeters wide. So we can see that this colony, and what we have actually is really cool microbes that we've labeled with light emitting portions. So we can stimulate them with a little bit of chemistry, and then they'll all go off. So we can actually count them in space by kind of turning them on at a given moment in time. We don't want to do it too often, though, because they consume a lot of oxygen when they do that. So that's another factor. But it's pretty exciting to be able to actually watch the development of this microbial colony in space and time. Now, what we see then on the left is that this small colony with flow going down slowly spreads out, and it's basically chasing the food. So the colony is actually growing upstream and laterally. And when it's doing that, it's putting out its polysaccharide kind of materials, which then fill the fine pores. So basically, the microbes are able to make the sand finer textured looking. And so it holds onto the water more, and it actually makes a little capillary barrier around its growing colony. In our models, we can simulate that same process and see the spreading of that colony. Okay, so the first thing we say is that, wow, microbes influence water flow. Second thing is it's a dynamic relationship where they're actually influencing water flow, perhaps and then that new water flow feeds back on where the colony will go. So it's a quite complex dynamic process. Now, we can also look at the water content impact. So that colony, as I mentioned, it's putting out these polysaccharides, which are basically surfactants reducing the surface tension of the water, meaning that it drains the soil below where the microbial colonization is highest. And so here, again, experimental and modeled systems showing that there's a water table which is about 15 centimeters below where the colony was growing. And you can see that ever darker middle portion, which is showing the decrease in moisture content because the microbes have actually dropped the surface tension of water, which made the soil drain. So what we see then, this is with one species of microbe. In soils, of course, we have on the order of hundreds of millions of species of microbes in a very complex web of interacting behaviors. So when we talk about the characteristics of soils, and we characterize their ability to hold on to water or to permit water flow, we have to keep in mind we're actually looking at a complex biological and physical system. Here we've talked primarily about the influence in a homogeneous media of the little cells and their exudates. But we also have to keep in mind that these same cells are going to create structures within the soil, PEDs and clods, which will create cracks and fissures. And so when we describe soils and the flow through those soils, we're not talking just about a physical system. It's a complex biophysical system where we're really looking at the ensemble behavior of a really beautiful and exciting, interesting, complex, and challenging process.